In less than 2 months since it was discovered, the Omicron variant of SARS-CoV-2 has become the dominant variant of the virus, causing an unprecedented rise in the number of cases of COVID-19 in the U.S. and elsewhere. The emergence of this variant has quickly led to some surprising claims about diagnostic testing for Omicron and a renewed appreciation of the importance of sequencing the viral genome for typing purposes. We'll discuss several questions about testing for Omicron, including, first, are rapid antigen tests sensitive for detection of Omicron? And should people swab their throats to increase the sensitivity of these tests? Second, how does the emergence of Omicron change our use of polymerase chain reaction tests for SARS-CoV-2? And third, how can we definitively identify the Omicron variant, and do we have the needed capacity for this? Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter at jclinmicro. I'm your host, JCM Editor-in-Chief Alex McAdam. This podcast is supported by the American Society for Microbiology, which publishes JCM. Please subscribe to the podcast and leave a rating or review wherever you get podcasts. I'm happy to be joined by my co-host and a fellow editor of JCM, Dr. Ellie Thiel. Ellie, it's great to see you.
Good to see you too.
We are also joined by two guests to discuss testing and sequencing of SARS-CoV-2. First, a frequent guest and also an editor of JCM, Dr. Melissa Miller. Welcome, Melissa. Can you introduce yourself?
Yes, it's good to be back. I'm at the University of North Carolina, where I am the director of the Clinical Microbiology and Molecular Microbiology Laboratories.
Thank you. We're also joined by first-time guest, Dr. S. Wesley Long from Houston Methodist Hospital and Weill Cornell Medicine. Thanks for joining us, Wesley. And can you tell us what you do at Houston Methodist?
I'm glad to be here, Alex. I'm the medical director of diagnostic microbiology at Houston Methodist.
Terrific. Thank you. Melissa, we're going to start with you, and thank you for emailing me an article about 15 minutes before the podcast came on.
You're welcome.
For my quick review.
It's coming quick and fast.
It is coming quick and fast. So we're all kind of on the fly here. There's a lot going on with Omicron. Information's coming in all the time. So let's jump right into an area that's been very hot lately, and that is rapid antigen tests for Omicron. Tell us what you know about the sensitivity of these rapid antigen tests for Omicron compared to earlier variants like Delta.
Yeah, I think when we think about sensitivity, we need to separate it into two boxes, right? We have analytical sensitivity and we have clinical sensitivity, right? And so different people think about sensitivity different ways. Like laboratorians, we start thinking about analytical sensitivity. But when my ID physician asks me, well, how sensitive is the rapid antigen test? They want to know clinical sensitivity, how well this is going to pick up disease, right? So what we know, and we'll provide the links to these articles, very early data show there was a paper in JCM, a commentary in JCM, showing comparison of 10 lateral flow assays. There's also data on the FDA website showing when you look at concentrations of virus and you compare Omicron with previous variants, and specifically Delta, that the level of detection is about the same. So Omicron is detected by the rapid antigen tests. Many of them perform very similarly at that same viral level. What's different, and we do have some indications, some of them anecdotal, in our own institutions. There's also a paper. This is Schramm et al. It's a preprint, and we'll provide that link as well, looking at PCR versus an antigen test during an Omicron surge. So what we know is that you can have false negatives with rapid antigen tests early in the disease, even symptomatic disease for Omicron. This is likely more to do with the disease course, right? With Delta, other variants, it was 5 days, 7 days before symptom onset after an initial exposure. With Omicron, we're seeing it more at 3 days. It's very quick. So even though a person is symptomatic and tests negative by an antigen test, there's virus there. You just can't detect it yet by an antigen test, but you would detect it by PCR. There are a few studies and anecdotes. Again, the plural of anecdote is anecdotes, not data.
Is not data.
It is definitely not data. The Twitter anecdotes are definitely not data. But, we are seeing some experiences in our own lab, even of some false negative PCRs early on. And I think there are a variety of reasons why you might see some of these. Some of them are specimen collection, time of symptom onset, whether the patient's symptomatic or asymptomatic, whether they're vaccinated or not. Now we have so many different variables that are feeding into test accuracy, it can be very difficult to tease them out. So recommendation really is when using rapid antigen tests, particularly at home, I always tell people, well, right now in this Omicron surge, if you're symptomatic, you have COVID until proven otherwise. And what I mean by proven otherwise is you serially take another or more antigen test, or you go get a PCR test. The challenge is, right, test capacity and availability of antigen tests and all of these other things. So we have to be careful. We still need scientific data. We still need to study this very carefully, but there are some signals that perhaps early on rapid antigen tests might miss some, not all, Omicron infections.
Perfect. Thank you. So let's turn away from antigen tests, which you all know I like to do, and talk about PCR tests. So what do you think about sensitivity of PCR tests? Any concern there with Omicron?
Again, we need systematic data. What we know is, and we have looked at our own data, is, again, that limit of detection, that analytical sensitivity is the same as that we've seen with other variants, right? The biggest concern has been about, because there's so many mutations in Omicron, are we going to miss the variant altogether, right? And FDA provides very good data and feedback and updates their website very frequently on what they know about differences and sequences and how they impact different tests, whether it's antigen-based. There's a couple of antigens actually that were on the website that we know don't detect Omicron. And fortunately for the PCRs, right, these are multi-target. So even if the S gene is included in the PCR, there are other targets, E gene, RDRP, other genes that are included because we knew that was, a risk. We purposely avoided tests, whether it was our own test or commercial test, that had an S gene. It turns out I'm kind of kicking myself about that now. Because it seemed obvious at the time, well, that's the one that's going to drop out. I don't want the S gene. We have one that has the S gene in it, but it's one that you can't see the differential results for the genes. You just get a positive or negative, right? So these tests that have the S gene in it, you've heard S gene target failures. TACPath is one on the FDA website that has been recognized as having this. That was also true for Alpha, right? Wasn't true for Delta, but we knew in the time of Alpha and now in the time of Omicron, if you have this S gene target failure, which means of the targets it detects, S gene is negative, the other targets are positive, it's called positive. So it's not a false negative result. But if you look at this differential detection among the targets, it gives you a clue that it's Omicron, right? Because the only other thing circulating right now, we'll learn much more about this later from Wesley, is Delta, at least here, Delta and Omicron. So we don't have Alpha. And so really, if you have an S gene target failure, it's Omicron, right? So you've got that kind of quick, easy detection. This is Omicron. Well, me, I didn't have that. So we had to wait until we had our sequence data to confirm that Omicron was here and had taken over Delta. So we don't have completely missing the variant, but we have target failures, and that can actually be helpful from an epidemiologic perspective.
It seems like it's become kind of a sales point for the companies that have S gene dropout. They're spinning it in a very positive way, saying this is a way that you can identify Omicron. Something that we would have seen as a failure in the past is now a benefit. Right. And it is a benefit, I think, a real one. It's also become, in the preprints and some of the things that we see coming through JCM, S gene dropout has really become a surrogate measure for Omicron, just as you said, Melissa.
Agreed.
Yeah.
Whether that remains to be true as, we get more evolution of the virus, we'll have to wait and see. But so far, we've kind of had these defined waves interspersed with waves that didn't have an S gene failure. So it has worked, but that might not always be the case. So,
And you touched on this a little bit, but let's go into specimen collection, because there has been a lot of talk about this, whether additional specimens, ones that we've not used much in the past, should be collected to increase sensitivity for Omicron. And I have to say here again, a lot of the data seem to be coming from Twitter, although we are starting to get some preprints on this. So what's your take on this? Do we need to start swabbing other body sites?
Yeah, before I dive into that, I saw a new word on Twitter last night.
Yeah.
Anecdata. It's the first time I've seen this. Anecdata. So from Twitter, instead of anecdotes or data, it's anecdata. So the data started to compile from all these different anecdotes, right? And I think that's really very applicable to this idea of should we be swabbing throats instead of noses? And really in the context of rapid antigen, this becomes a little tricky, and I'll talk about that. But, early in the pandemic, we will all remember back 2 years ago. It's very, very hard to think that we've been doing this for 2 years. We were in this boat. We were trying to figure out what were the best specimens. Was it NP? Was it OP? Throat swabs? Was it saliva? We validated all of these. We got saliva from our drive-through clinics. And I've said before, I think maybe even on this podcast, like if you would have told me, 5 years ago, I would have been validating saliva for a respiratory virus, I'd have been like, no, you're lying. This is crazy. So here we are, right? So we know with SARS-CoV-2 that you can readily detect high quantities of virus in saliva, especially early in disease. And even pre-symptomatic patients. So the challenges can be, specimen collection. There's different ways to collect saliva. We've actually addressed this on a previous podcast. The cough and the spit, or just the spit and all of this kind of stuff. Now, I bet everyone listening has their mouths watering now. You're welcome. Oops, sorry. Siri's mouth is watering too. So we know this is really an acceptable specimens for SARS-CoV-2. So now the question has really turned, is it a better specimen for Omicron? And so far, I've only seen one preprint that actually addresses this. And this one is, sorry, I just want to make sure I get the right one, is the Marias et al. Saliva swabs are the preferred sample for Omicron detection. Again, this is a preprint, not yet peer-reviewed. So in this particular paper, they used a composite standard, and they were looking at saliva swabs and mid-turbinate swabs against this standard. And what they found was when you looked at it with the Delta variant, it wasn't superior. However, when you looked at it with the Omicron variant, saliva and mid-turbinate swabs had 100%, saliva 100%, mid-turbinate 86% sensitivity against the standards. So there are some data, relatively small numbers still, but to suggest maybe you see replication of the virus in the throat and therefore detected in saliva early in Omicron. Again, we need more data, systematic data to look at this. What does this mean? Is this a component of the virus and mutations and immune response? Lots of things to look at regarding this. I think probably the most common question I've been answering this week is throat swabs and rapid antigen tests, right? Yeah. You see these pictures on social media. I swab my throat. I swab my nose. It's negative. I swab my throat. It's positive. I can't help but think back to the pictures we saw. I think it was Matt Beneker. I think maybe he did Coke, and maybe Jen Dian-Bard did coffee. And I think that's right. I think that's who did which one. But where you can take these, acidic, kind of liquids, put them through a rapid antigen test, and get a very nice positive line. Well, think about all that passes through your throat and the environment in your throat and the timing and how you collect it. Well, we need to understand how that impacts these rapid antigen tests, right? So absolutely, I completely support the statements by FDA. You should not be using these off-label. I know a number of people have also pointed out it's not easy to collect a throat swab if you do it right. No. Right? You might think you're swabbing your throat. If you're not like gagging and about to throw up, you're probably not swabbing your throat correctly, right? So that's the other concern. And we have to remember, like with home tests, part of the FDA's job is to keep the public safe. We all want that, right? But it's not only about, okay, is this the absolutely best specimen for this test? We have to think about people swabbing themselves and what's safe and what could go wrong and the type of swab to be in the kit. That's all part of this that I think a lot of times that's a nuance that the public doesn't appreciate, right? So I fully expect to see that we might see greater sensitivity. We might see data that shows greater sensitivity for throat swabs or even saliva, but that doesn't mean they should be used in at-home tests.
I'm not quite sold yet. I think it's possible that we're going to see higher sensitivity with throat swabs or saliva. But I guess I'm not there yet. And also I would like to see it compared to a nasopharyngeal swab, which I guess makes me very much old guard.
I will say, and any of you who have listened repeatedly to me being on these podcasts or read things I've said in the media, you've probably noticed I have evolved in 2 years. A year and a half ago, I would have never said we should be doing at-home antigen tests. I might have even said it on this podcast. I don't remember. At that time, we didn't know enough. I'm a data-driven person. We all are on this podcast, so I want to see the data, and it's challenging in this environment. I mean, we're in a crisis. We're still in a crisis, right? And what's good enough? I am not supporting perfect anymore. The perfect send to a lab, do a PCR, even though we all know that's not perfect, but, that's kind of our reference method. An NP swab or NPOP probably would be the reference method sent to a lab. So why is that, you might ask? I see the people. I see the people in my lab, and I see how stressed they are, and I see how broken they are. And I see us vying for, like I told our supervisor just earlier this week, we might be taking a road trip to Illinois to pick up some reagents, right? I was kidding. At least I hope I was kidding. But I mean, it's just, we're in this complete crisis of not being able to manage the test, right? The testing is just a small piece of this whole thing, but not being able to manage it. So how can we get more people tested that is safe for them, safe for the public, but still try to keep our infrastructure in the lab in place? So I definitely think, at least with this Omicron wave, we absolutely need to be doing at-home testing.
Yeah, I'm with you. I wouldn't say I've evolved, but I think I've budged. I used to be very sort of, I won't say anti-rapid test, but I did not like the direction that people were going in terms of the utility of rapid antigen tests. But that said, when I say that I'm not sold on the throat story, I don't mean that I'm not sold on rapid antigen tests. I agree with you. They definitely have an important place, and I used a couple of them the other day myself. So I'm with you. Ellie, what's your take?
Well, my take is I can't find a rapid antigen test to save my life. There are not a single rapid antigen test in Rochester, Minnesota. I do think they will be playing an increasing role because the central lab testing situation right now is not sustainable. So one question I actually have for the group is, should we be considering maybe amending who gets tested in a way? For example, we're still testing close contacts of fully vaccinated asymptomatic individuals that are close contacts. Should we be doing that right now? So should we be rethinking some of the individuals that we're testing? And
we're not testing those people.
Now?
We had to stop just in the last couple of weeks. We stopped, through our drive-through clinic that's here on campus, asymptomatic testing outside of very select pre-procedure testing, high-risk pre-procedure testing, and some employees. We also, even though the tests aren't coming to us, like our outpatient clinics, they can't send us asymptomatic testing anymore. They have to send to you, to a reference lab like Bayo or Quest. So I'm part of the problem. But I mean, that just shows the complexity, right, of this world, right? So I'm doing that, but you're still taking my tests, right, that are asymptomatic tests, for example. So, so I will say, actually, ASM and IDSA are thinking right along these lines, and can we put together some guidance of prioritization? We're not going to tell people what to do and who to test, but in times of incredibly high testing demand, and your point about not being able to find a rapid antigen test is an excellent point, right? I guess my statements were solely based upon the idea that everybody had access to these tests and that they're affordable, right? I mean, it's just, they're crazy expensive. And you're telling people to test over and over and over, and you have a family of 4 doing this that are exposed. So I don't think it'll necessarily, and God, I hope we're not talking about this in a year, but become the testing paradigm for this pandemic. But I do hope, like, what we've learned testing wise can be carried forward into future pandemics. So that's all I can hope.
Well, the guidance you mentioned sounds like it would make a fine commentary for JCM, and I look forward to seeing that. Wesley, what's been the approach with asymptomatic testing in Houston Methodist?
So the biggest change we probably did to cut down on testing of asymptomatics was we did have we had been doing employee screening weekly or biweekly, depending on sort of tiered risk, via PCR, via walk-in clinic, and in December that very rapidly started to get overwhelmed. And some people seeking testing before the holidays or before gatherings. And so one of the shifts that was made was the hospital system actually acquired a large number of rapid antigen tests and shifted to a system of, if you're asymptomatic, here's a take-home rapid antigen test. If you have symptoms, please go ahead and go get the PCR to try to basically decompress some of the pressure on the PCR testing. I think otherwise for the actual testing of patients that we're doing, we haven't made too many other changes. It's basically if we get a PCR test, we get the order, we do the test. There's certainly, some through the whole pandemic, we've been prioritizing certain tests that need rapid turnaround, transplant patients, laboring mothers, those sorts of things, to faster PCR tests. But that's really the extent of the sort of throttling and adjusting we've done for asymptomatic testing.
Thank you. And I just want to do a quick round robin because a couple of us have mentioned the stress that labs are under. And I'm not going to ask whether your lab is under stress, and I'm not going to ask you to describe it because I'm sure your lab is under stress, just like mine is. All labs are under absolutely tremendous stress. So what I'm going to ask instead is, has your institution done anything recently to alleviate that stress? We did. Children's created a labor pool where non-clinical people can volunteer to go and lend a hand doing appropriate tasks in occupational health and in the laboratories. We have them labeling tubes and that kind of thing. And it's been extremely helpful. It's really relieved a lot of the pressure on the lab. So any tips that you guys can share? Any things your institution's done? Why don't we start with Melissa?
Yeah, we have some temp techs that we were able to get that we've had kind of in different contracts during the 2 years. So we have 2 temp techs now. Part of that helps us to cover weekends. So it gives, even though all of our people rotate all the weekends, we're 24/7. It gives them at least a shift break that our core molecular staff gets a little bit more time on the weekends. So the 2 newest ones actually have just finished training and going to their shifts. So I think we'll see a big impact there. I like that idea, Alex. We have what we call helping hands and surveys go out and people sign up, but it has largely been clinical, clerical work, collection sites, traffic, at the drive-through clinic, because there was a shortage at the collection site for a long time, and people, right? This is a people issue now. I mean, we do have some supply issues, but it's really a people issue. Just not having enough people being out with COVID, and on their isolation period, and some people at 5 days don't feel good enough to come back. So test or not, they don't feel good enough to come back at 5 days. So, and I will say, just, it sounds, maybe trivial, but we get lunches sometimes, just our president just this week stopped one of our managers in the hallway and says, I know how much stress you guys are under. Buy lunch for the lab, so it's little things like that. I get notes all the time from people. I always forward them to the lab, and they really just are amazing. I mean, I wish there was so much more we could do for the techs.
Yeah, amen. I agree.
Mm-hmm.
Ellie, anything that you've tried in your lab or that your institution has tried?
Yeah, so we just recently set up a similar volunteer pool for our molecular area for COVID testing. And, it's so uplifting and encouraging when you see, over 100 people volunteer to help process samples, basically. So that's one thing. We also get lunch and cookies, which it helps boost morale too. And then our department has a joy committee that comes up with various 15-minute activities that you can do to just get your mind off of the day-to-day. So our department has definitely attempted to step up and help.
Awesome. And how about you, Wesley?
We've had a very similar labor pool situation throughout the pandemic. It's honestly probably been more helpful on the research sequencing side and helping with clerical tasks there. It's helped some in the clinical lab early on. It hasn't been as useful for us in Omicron for whatever reason. And then, there's been food motivation, food donations. Ironically, the summer is supposed to be the summer of rest and recharge, which Delta sort of put the kibosh on. But every employee, we had $500 gift cards for Christmas from a local grocer and things of that nature. So it has been stressful, but definitely things like labor pool and things, different sort of motivators and just morale boosters have been helpful as well. They've also started, I just had to put in a plug, we started a free mental health service, essentially in-house, for all employees to seek counseling and other resources that's opened up just in the past couple of months. So that's been helpful for those who are really struggling to have that available.
Yeah, I'm glad you mentioned that. I'm certainly no expert on this, but people should be taking advantage of that kind of resource, 100% if they need it. I want to turn now to a couple other subjects. Ellie, tell us, you're the serology maven. Tell us whether there are any consequences to the changes in Omicron with regards to serological testing.
Yeah, so there have been some questions as to when the current serologic assays will pick up antibodies to Omicron. And just generally speaking, despite the high number of mutations in the spike protein in particular, the qualitative assays for both anti-spike and anti-nucleocapsid, because they're detecting polyclonal antibodies, there's still enough shared epitopes between the Omicron spike and nucleocapsid versus the ancestral variants that the sensitivity, the impact on sensitivity would be pretty minimal. I think the question currently is more whether the quantitative serologic anti-spike antibody assays and the surrogate neutralization assays would be impacted more, since these assays, again, are based on ancestral proteins, and they may not pick up all of the antibodies that are developed against the mutated epitopes. And so that could potentially throw off the quantitative results. But, it's kind of challenging to look at that because you need to get serum from patients with Omicron collected 7 to 10 days after infection, and they have to be unvaccinated. So that's kind of a lot to ask for. So it's the data is still out at this point. Overall, though, regardless of the circulating variant, the role of serologic testing hasn't really changed much at this point. So I think it's kind of a moot issue at the moment.
Gotcha.
But we'll see.
I think it will be interesting to see, especially in those surrogate neutralization assays, whether there are changes. But as you pointed out, it's going to be hard to get specimens to do those studies.
Yeah.
So let's turn to sequencing. And Wesley, your lab has been extremely active. And in fact, you have a couple of preprints up and you keep updating them, which is kind of an interesting publication model from an editor-in-chief's perspective. I kind of love it. But your lab has a history of sequencing. So when this all hit, you were ready to go. What was your lab doing? Why were you so well positioned as you were?
So we have in the department, in our research division, we have a very long track record of sequencing large population-based sets of bacterial sequence, data, generating data, and that, typically in a retrospective fashion. So maybe we would take several thousand Group A strep or Staph aureus or Klebsiella and sequence all of those, and then go back and dig into the patient data and look for mutations associated with phenotype and those sorts of things. So we had sequencers in-house that were used both for clinical molecular oncology, which at the beginning of the pandemic all started being sent out so they could focus on COVID testing, and then the expertise in the laboratory to do the sequencing itself. So we had that pre-existing infrastructure, and we just had to pivot really in January of 2020 to, okay, now we're going to focus on this virus.
Gotcha. And what has been the scope of this sequencing project for SARS-CoV-2?
So we really, from day one, from the day it was declared a pandemic, the chair basically told us, like, we are going to make the good faith attempt to sequence every single positive sample that we isolate from a patient in our hospital system. So it's an 8-hospital system with a variety of standalone EDs, and we get private clinic business and other things. And so from day one, the goal was we're going to sequence everything because it's the only way to have this sort of large, comprehensive population-based data set. And so that was the scope that we were charged with. And then, of course, as the pandemic went on, obviously more and more positives, there were a lot of adjustments had to be made, but that was from day one, we were going to, attempt to sequence everything. And that's been what we've done through the pandemic.
Do you have to use a CT cutoff for low viral load specimens that may not be able to be successfully sequenced?
So we do not. And the reasons are sort of multifold. One, and we really agonized about this early on, was our data set being biased because we were getting better quality sequence out of the lower CT samples than the higher CT samples, and we dug into that for a while to reassure ourselves. But the other issue was that to meet our clinical needs, we ultimately wound up using 4 or 5 different commercial platforms, and not all of those platforms would even give us a CT. One gives fluorescent units. Some of the platforms don't give us any sort of indication about the CT at all. So in order to basically sequence everything, we never put in a cutoff. It was just, we're going to make the attempt to sequence everything. If the sequence fails, it fails, but we'll get the best quality sequence we can from every sample.
And what's the method of sequencing that you use? Is there anything specific you've had to do besides add a reverse transcription step to pivot to SARS-CoV-2?
So I have to say what I did very early on is I was charged with figuring out how we were going to sequence it. Was I found the Arctic group and the Arctic protocol. So there was already a pre-existing protocol for amplicon tiling the whole virus. They had pre-baked bioinformatics pipelines in Docker containers and things that I could download. And it was all based on Oxford Nanopore. And we had an Oxford Nanopore GridION sequencer in-house. So we had familiarity with Oxford Nanopore. We had the hardware for that as well. So we started out using Arctic, using the Oxford nanopore, and that was how we sequenced really into the summer of 2020. Then, as volumes kept increasing, and we couldn't keep up, we added a second GridION, and we couldn't keep up. That's when we made the move to switch from using Oxford nanopore to using Illumina, because we had Illumina instrumentation that was higher throughput, and then ultimately acquired even larger Illumina instrumentation that was even higher throughput. So we could do batches of 768 specimens at a time instead of 24 on a flow cell on the Oxford nanopore. So we adapted. We used actually some other people adapted, and we adopted the Arctic protocol to using Illumina sequencing that we were familiar with, and charged toward using that. And that's what we're doing still today is Illumina. So we've made some adjustments and some changes. We've added a tremendous amount of automation. So in the beginning, it was very, very manual and a lot of multi-channel pipetters, and now we've got about to be 3 biomechs to do fluid handling. We've got these little things called some mantis, and it moves a pipette tit around and spots things in wells. It's very cute and fast. But we've added a bunch of different pieces of automation hardware. We've also added personnel. So in the beginning, I think we had really 3 research technologists working on it, and now we have a team of 10. And so it's really, we've added people and machines, and it's been a tremendous effort.
So you've had to really scale up and evolve to keep up. But Melissa, yes. I know you have or work with a sequencing program as well. How do your experiences compare to Wesley's?
Yeah, I was sitting here thinking, oh man, that's a whole other beast than what we're doing. But thank God for people like him. So, yeah. I think, our model is very different, but really shows how clinical micro labs that weren't doing kind of surveillance sequencing before can get into the game. We were not sequencing in 2020. We started in January 2021. I think through 2020, we were just trying to keep our head above water. And we kept thinking, oh, surely, other people are going to do this. Public health labs are going to do this. Academic centers are going to do this. And there weren't enough sequences, especially for North Carolina. So we started early in 2021 working with our genomics core on campus and formed a relationship with them. I'm not a bioinformatician. I don't want to be a bioinformatician, although it might be a second career path. So just a really wonderful group that... we provided the specimens, the metadata. They did a lot of also Arctic primers on a GridION. Our volume is much lower, Wesley, thank goodness. So, and we're not always sequencing all of them, right? So realizing that introduces bias in it. We do have a CT cutoff, and that actually changes depending on our volume, right? So sometimes we can sequence all of them, and we try to sequence all of them independent of CT values. Other times we're like, oh, we have way too many specimens. We're going to take only 30 or less or 25 or less in terms of CT values. Actually, there's a commentary. I may be just out in JCM. Kyle Rodino and I are the lead clinical microbiologists that describe this academic clinical lab partnership at Penn and here at UNC. So in the summer, we transitioned the sequencing actually to the clinical lab. And our microfellows were involved. I ended up hiring, again, research personnel to work on this, but still in collaboration with the genomics core. And now the state actually funds this work. So different scale, but shows it's doable. We're all still nanopore. It's been quite a ride.
Oh, yeah. And I think that manuscript is in the recently accepted bin on JCM's website, but we'll put a link to it with the show notes. Wesley, this is a big question, and you're going to have to pick it apart a little bit, but can you give us an overview of the results of your sequencing program? What things have you found?
So, I guess we're talking about Omicron. In the very beginning, one thing we found was that we had sort of multiple, almost simultaneous introductions into Houston of COVID-19 strains from sort of different places around the world. So that made us different than New York and Seattle. Then, going through now to the present day with Omicron, I think the biggest finding with our latest preprint, which we've updated 3 times, has been, a lot. It has matched somewhat what has been seen elsewhere, but that we're seeing, a significant increase in breakthrough infections with Omicron compared to Delta, compared to Alpha before that. so there's an increase in breakthrough, but along with that, we're seeing, on average, a decrease in severity, a decrease in hospital admissions, and a decrease in length of stay, and sort of measures of severity like needing mechanical ventilation or needing things above room air. Those are the major findings we've had. Now, our hospital system, to an extent, we are undersampling pediatrics because we don't do pediatrics here at the main mothership. We do some pediatrics at some of the outlying system hospitals, but we're not Texas Children's. We're not a big pediatric center. So, but even still, there's a lot of things in the data that we're still picking apart and still adding to, looking at, why it is we're seeing less severity, and how much of that perhaps is due to the virus, and how much of that is due to the fact that we're seeing breakthrough cases in people that are vaccinated. And on average, our Omicron patients are younger and healthier than the patients that we saw with Delta. And so that may be another reason why the severity seems less. So there's so many different things that figure into it that it's a difficult, we also have to try to temper the message of, on average, it's less severe, but that doesn't mean that for an individual that their case is necessarily going to be, I hate to say the word mild, or less severe than it might have been with Delta, because we are still seeing some patients with severe disease that have Omicron, and it's just there's fewer of them. The other problem, of course, is that there are so many Omicron patients that even with all the parameters I've given you, it's still led to an increase in hospitalizations. It's still very stressful for the ERs and everything else. Just to have so many patients, that's been a huge stressor, I think, on healthcare systems across the country.
Yeah, I think those are very important points. I'm sorry, Melissa, please go ahead.
What's like the turnaround time of your sequencing? I'm just curious, because we were being really pressured, right? Like headed into Omicron, not having an S gene target failure. How quickly can you tell us? So I'm just curious if kind of like what you've set up there, which is this total enterprise. It's really impressive.
Right. Thank you.
Can you use this clinically? Like to make these? Is it fast enough? Is what I'm asking, to make any sort of clinical decision.
So I do want to say just very quickly, and I do have to give great thanks, of course, to the support of the department and the hospital system. Really, all of our support is internal. So we haven't gotten any money from the state. We haven't gotten any money from federal government. It's all internal support, some philanthropists in Houston. It's all hospital money running the show. But
wow.
So true. Yeah, it's tremendous. Yeah,
wow.
Hiring. I personally, in the city of Houston, is incredibly blessed to have this effort here. But in terms of, and now I'm blanking on, I did my plug and I blanked on your question.
Turnaround time, like how quickly you get results. Yeah.
And clinical utility.
And clinical utility. So unfortunately, it does take time to do the sequencing process. And so we're always, we have this big team, and there are always different plates. Every day we have plates at different stages, but it's about as fast as we could ever get it. Was just a little bit over a week, so maybe 8 days or so, to go from turning positive to getting the sequence and getting it analyzed. And we have, in the beginning, I was doing analysis. Now I have collaborators at the Patrick Resource in Chicago that do the bioinformatics, a lot of the bioinformatics. We do a lot of the back end analysis, but so that's not really clinically useful because it's 8 days. The other thing is the FDA is very clear that research sequencing can't be reported back to patients, to physicians. So we've never done that. And so, funnily enough, talking about SGTF and PCR and target failure, so we were charged, other people, Dr. Randy Olson in our molecular lab was charged in early December with bringing a SGTF PCR online so that we could set up sort of a dual path where positives would be, because we had been taking positives and sending them to, actually taking everything and sending it to research and pulling positives, but to basically also then take those positive PCRs from our clinical workflow, take a sample for the SGTF assay, and then report that assay onto the chart because it is a clinically validated assay done in our clinical lab. So the SGTF PCR is done in the clinical lab. It's a clinical assay, and the sequencing is still all research. But so we had to do that to have something clinically useful. We had to bring on SGTF. And, the irony is that now we're at 97% Omicron, but still, We may be identifying, they're starting to use it. So now the pharmacy is starting to use it when they're scheduling people for sotrovimab and for Evishield, et cetera, to make sure, like, if we're scheduling somebody for sotrovimab, let's make sure they don't have Delta, because we might still have some Regeneron around that they might be able to use or something like that. So they've started to use the SGTF PCR clinically, but the sequencing, the turnaround was just too slow for that.
So
I
I
want to
picked
compare
up
to your practice, Melissa, but just before we do that, so am I right, Wesley, you are reflexing all positive PCRs to a separate assay for that S gene failure as a surrogate measure for Omicron?
Yes.
And then sequencing as well? Man, you got
Yes.
resources.
Yes.
Well, I was going to ask you, you said all were going to sequencing at some point. Does that mean you were sequencing the negatives at some point too?
Oh, no, sorry. So, I mean, we hopefully not.
Okay. Well, no, I mean, some people argue, right? We're totally biased with our diagnostic test. If we don't
Right.
sequence some proportion of negatives, how are we going to know our diagnostic tests aren't missing something, right?
I did make a pitch. I did make a pitch in the trough. I made a pitch in early November before we knew Omicron was going to happen. What if we sequenced some of our negative respiratory panels, where we have ostensibly a patient with symptoms who's negative for everything on a respiratory panel? What if we just did sort of metagenomics on those? I didn't get a lot of traction for that. And then Omicron happened and we were off to the races. But no, I was only saying all of the specimens, just to make another pitch for how hardworking the research team is. Basically, these high throughput sequencing instruments or high throughput PCRs, we have racks of specimens, and then the research techs have to go through and scan to pull the positives out of the racks in the research lab to know which ones to sequence. So they have to do that work on top of doing the actual sequencing.
And this may be a slightly unfair question because neither of you has a direct role in this, but I bet you have opinions. And Ellie, jump in as well. But what are your thoughts about sort of national capacity for sequencing? There's been a lot of complaining about the level of national capacity. I and you know the folks at the CDC who are doing this and rolling this out through the regional public health laboratories, the state laboratories, and so on. And they're good people and they're working hard. But what are your thoughts about the national capacity? We'll start with Melissa.
I was pointing to the other square.
Oh, is that right? All right. We'll start with Wesley. We'll come back to you.
Yeah. I think the capacity has gotten better in 2021. It was pretty sad in 2020. There wasn't a lot of sequencing being done by public health. There weren't a lot of resources dedicated to it. Then things changed in 2021, and suddenly there was a lot of public health directed sequencing, a lot of it in partnership with large reference labs. So that was good. One of my complaints about a lot of the public health sequencing is they strip the data down to just the state level. I'm a big proponent of at least that we don't, on our samples, we only report that they're coming from Houston. We don't get down to zip codes and things because it's potential identifiers. But I do wish that from public health, I was getting data with city coded to it so that we could do a little bit more sort of comparing our samples to other samples from Houston. I mean, now if it's coming from Harris County Public Health, then I know. But there are some, state public health or CDC. We really have no way of knowing other than it's in Texas, which is a very big place, where those isolates are coming from. So that's my one complaint. I think 2021 has been better. I do think the UK still blows us out of the water. And I think if there's whatever lessons we could learn from their efforts and try to replicate here on a national level, I think it would benefit us in sort of future not just future pandemics, but just future disease surveillance in general. To have a better infrastructure for national sequencing capacity, I think is important.
Yeah. Melissa?
I couldn't have said it any better. Definitely not. I mean, I think that woefully unprepared for this pandemic on so many levels, but the genomic sequencing, without a doubt, wasn't even close to what we need. So again, just trying to get back to some level of optimism, which keeps me going day to day. If you're not optimistic, I don't know how you would survive right now. But how can we use what we've learned and what we were missing so this never happens again, right? So if we put this system in place, including academic labs, research, clinical labs, it has to be beyond the public health labs, right? That's not enough infrastructure. Of course, this all costs a lot of money too, right? But we can't let these instruments, these things that we bought to do this sequencing, gather dust when this is over. Hopefully it'll be over soon. But so what do we do next? How do we as a network of people who have started sequencing continue this work and make sure we're sitting on ready for the next pandemic? I think that's what our focus needs to be, when we're out of the big umbrella of COVID.
I agree. And I think that optimistic note is the perfect place to wrap this up. Ellie, terrific to see you. Hope you're doing well.
Thanks. Good to see both of you. Thanks for your perspectives.
Melissa, thanks again for joining us. I'm sure we'll have you back again soon.
Sounds good. Thanks for having me.
Yep. And Wesley, thanks very much for joining us. I'm sure we'll want to have you back as well.
My pleasure, Alex. Thanks. Anytime.
And thanks everyone for listening.